All right, guys, I'm ready to get started with section 2 of our genetics unit. Again, guys, this is pretty exciting stuff. I enjoy going over genetics. A lot of genetics principles you can relate to, such as these traits we've been talking about. Really quickly, I want to go over the last slide from the last lecture, and that was monohybrid crosses. But before we do that, let me remind you that this is a self-paced unit, that you need to continue to work very hard on this, that if you get finished with all lectures and assignments, you are welcome to take the test anytime after next Monday. I think that's the 13th. And if you complete that before next Friday, then you'll have some days to yourself. The only thing that has to be done is you have to complete everything and be ready to take the test by next Friday. All right, let's get started. So remember, monohybrid crosses, we're looking at one gene or one allele, one gene, but two alleles from each parent, obviously. And when those two combine, we put one on top, so big B, little b, that's heterozygous. And then the other parent, big B, little b, again heterozygous. And then we just drag and drop these like so. I can kind of show you here. So this would go here, here. Then this would also go here. Little b would go here. Big B goes here. Little b goes here. And then little b goes here. And little b goes here. So we end up with one homozygous dominant, two heterozygous, and one homozygous recessive genotypes. And those in turn, since this is heterozygous, this is homozygous dominant, both have dominant alleles, then we have 3/4 of these dogs being short hair, since that is the dominant trait, and then only 1/4 has long hair. All right, so now that we have reviewed that, let's start with some new content. Sorry, I'm just trying to make sure I don't make the same mistake I did. Trying to figure out how to do this real quick. Give me 1 second. All right, sorry about that. I figured it out. You should be able to see the entire screen now. So let's get started with the second section. Again, guys, this will not take nearly as long as that first part did. All right, so Gregor Mendel, he wanted, or had a question after he looked at these monohybrid crosses, and that question was, if during gamete formation and when these gametes, when these alleles segregated, for what he means, or reminder of what segregation is when egg and sperm fuse, you get one allele for each gene from a parent, so you get two alleles for each trait from total, but one from each parent. So when they segregate and they either have, a dominant allele and a recessive allele, homozygous dominant, homozygous recessive, he wondered if the segregation of one pair of alleles could have an effect on a second pair of alleles and another trait. So, for instance, does the gene for pea plant height, such as being tall or dwarf size, have an effect on the seed color? So he set this experiment up, and he followed, this time, instead of one gene, followed two genes. And this was known as a dihybrid cross. I'll never refer to it as a two-factor cross. You will always see me refer to this as a dihybrid cross. So he compared round seeds and wrinkled seeds, yellow seeds and green seeds. So one parent had peas that were round and yellow. As you can see the difference between round seed here, yellow seed here, or round seed here, wrinkled seed here. And then he crossed that with a parent that had wrinkled green seeds. And then he knew before doing this that round and yellow traits were dominant. So that means if there's a dominant allele for round, so big R, or a dominant allele for yellow, just one present, then it's going to be round and yellow. So first he crossed true breeding plants. Again, what true breeding plants are, guys, to reiterate this is one of your vocabulary words. It's just plants that form offspring with themselves. So the pollen from the male part or the stamen fertilizes the egg inside of the pistil on the same plant. So he did this, and he crossed round yellow seeds with wrinkled green peas. Round yellow peas with wrinkled green peas. Remember, these are the dominant trait, the round yellow. And what he assigned for the allele combinations were big R, big R, big Y, so homozygous dominant for both traits, and then homozygous recessive for both traits. And he knew that all F1 offspring had round yellow peas. So, if round and yellow are dominant, what is the genotype of all F1 offspring? It was heterozygous for both traits. And there is a way to determine this, and I'll show you that in just a second. But next, Mendel allowed the offspring, so the first generation offspring, to self-pollinate. And this is what he found. So when the first generation was allowed to self-pollinate, so we had big R, little r for round seed, so heterozygous for round seed, heterozygous for yellow seed, and we bred that with a heterozygous round, heterozygous yellow. It resulted in the production of 556 seeds. Here's the combinations. So we had 315 round yellow seeds that were dominant. He had, so what I mean by dominant, round is dominant, yellow is dominant. He had 105 round green seeds, round being dominant, green being recessive. 104 wrinkled yellow seeds, wrinkled being the recessive trait, yellow being the dominant trait, and 32 wrinkled green seeds, which were both recessive. And this meant something. The alleles for one gene had no effect on the alleles of a second trait. This was known as independent assortment. These alleles divide or are independently assorted and do not affect one another. So this brings us to the principle of independent assortment, which Gregor Mendel coined, and it states that when gametes are formed, the alleles of a gene for one trait segregate or separate independently of the alleles of another trait. So in this case, seed shape, for instance, round or wrinkled, didn't affect seed color, yellow or green. So let's look at how we actually produce a Punnett square for a dihybrid cross. A little bit more tricky, so please pay attention. This Punnett square will have 16 squares in it. Each parent passes one allele. That's important to remember. One allele of each gene pair to the offspring. So each parent will give the offspring one allele for seed shape and one allele for seed color. Giving the following parental genotypes, what alleles could each parent pass to their offspring? So if the parent was big A, little a, so heterozygous for both traits, whatever these two traits were, doesn't matter. We could call this height and seed color. And another parent was heterozygous for big A, or for whatever the A trait is, homozygous recessive for whatever the B trait is, And then we had 2 more parents. Here's how these would break down. So let me just kind of draw this for you, if I can. Hold on one second. All right. Now I think I'm ready. So big A will combine with both big B and little b. And then same thing with small a. It will combine with both big B and little b. So these would be our 4 allele combinations for this parent. And same goes for all of these other parents, okay? So let's look at an example. All right, so let's do this here. So we're going to do a true breeding round yellow with a true breeding wrinkled in green. So the genotype of each parent, so true breeding meaning that they're dominant for each trait. Give me one second. Sorry, my wife had a question. Anyways, let's get back to where we were. True breeding meaning they are have dominant, homozygous for both here. So homozygous dominant for the true breeding round and yellow, homozygous recessive for the true breeding wrinkled in green. So let's determine these allele combinations that can be passed to the offspring. Okay, again, we have our traits here. So let's do this way. All right. So we're going to go, our allele combinations are going to go, excuse me, big R, big R, big Y, big R, big Y. Then again, big R, big Y, big R, big Y. And then over here we're going to have, since they're both homozygous, we're just going to have small r, small y, small r, small y, small r, small y, small r, small y. And then we combine these. So big R first, always in front, and then little r, big Y, little y. Again, this is going to be the same for all 16, so I'm just going to go ahead, put this on there for you to look at. Already have drawn this for you. So we're going to have 16 of the exact same genotypes, heterozygous for round yellow seeds. All right. Next, what if we allowed those to self-pollinate? So meaning that we had those heterozygotes, big R, big Y, little y, and we combine those. So I want you to try to solve this on your paper before you move on. So pause the video, try to solve this by yourself. And I'm going to go ahead and write this for you. So we're going to go big R, big Y, big R, little y, little r, big Y, little r, little y. And then for the other, it's going to be the same exact genotype. So big R, whoops, big Y, big R, little y, little r, big Y. Again, the capital letters represent the dominant trait, okay? So don't get confused there. And then little r, little y, okay? And we're just going to combine these. So here we're going to have big R, big R, big Y, big Y. So there's a homozygous dominant for both round and yellow, okay? Then we're going to do this for all these guys, okay? So I'm not going to do all these by hand. I will switch over to the PowerPoint and do that. But here we have homozygous dominant for round seeds, but heterozygous for yellow seeds. But it's still going to be round and yellow because yellow is the dominant trait as opposed to green. So here, let's do one where we're going to have, let's do this recessive one here. Little r, little r. So little r here, little r there. So we just kind of draw it down like this. And then little y, little y. So there's a recessive, homozygous recessive genotype. So let's go back out of this and solve this ourselves. All right, so I'll do that for you. But let's go ahead and fill this out. Okay, so again, this is, well, let me get that off. This at the bottom would be, so just so you can see, it would be homozygous or heterozygous for round seeds, and then it would be homozygous recessive for green seeds. So here would be our genotypes. Let me go ahead and put this back big screen where you can see it a little better. All right, so let's mark these out real quick together. So big R, so homozygous dominant, there's one of those. Homozygous dominant for round seeds, heterozygous for yellow, there's two of those, 1, 2. This genotype, big R, little y, let's find those, big R, big R, big R. There's only one, okay? Big R, little r, big Y, big Y, big R, little r, big Y, big Y. Yep. Heterozygous all the way around. Boom, 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 and there, you can't hardly see it. Sorry, I don't know how to hide this. big R, little R, big Y, or little y, I'm sorry, there and there. Then we have there is this one, there, and last is 1/16. 1 out of 16 is recessive. Okay, and then of course we can look at the phenotypes. There we go. Have any questions, come see me, but I think this is pretty self-explanatory, okay? So now we can see the dihybrid cross and how that works. Last one, I want you to do this by yourself. Right-handedness is big R, dominant over left-handedness, little r. The ability to roll your tongue is the dominant trait over the inability to not roll your tongue. So it's asking, what offspring might be expected from a cross involving the following parents? So this is going to be a right-handed person that can't roll their tongue, and it's homozygous dominant for right-handedness, homozygous recessive for tongue rolling. And this is going to be a right-handed person that can roll their tongue, but it's going to be a heterozygous for tongue rolling. So go ahead and pause this video right now and do this on your own. Go. All right. Hopefully you paused the video and you have started the video back. So we're going to go over these answers real quick. First, we have to separate these into a little combinations. Remember, we go big R, T, big R, little t, big R, T, big R, little t. And then big R, big T, big R, little t, big R, big T, big R, little t. Okay. Then you combine these, you get this. You can count these up yourself. Best thing to do is count them off as you go. So look at one, write it down, then see if there's any more of those. So here's one of those, 2, 3, 4, 5, 6, 7, 8. Looks like 8 total that are big R, big T, little t. So 8 out of 16, I was right, half. And then half are big R, little t, little t. So half of them will be right-handed because it's homozygous dominant. And then they will be tongue rollers because it's heterozygous. Again, this is a dominant allele. So therefore, the principle of dominance and recessiveness says if there's one dominant allele, then it will mask the recessive trait. Here you're going to have, again, homozygous dominant for right-handedness, but homozygous recessive for non-tongue roller. So half will be right-handed and a non-roller. And I was wrong. Got one more. You can pause this if you need to finish writing that down. We have one more. So go ahead and pause this and do this on your own really quickly. All right, we're back. So it says a woman who is right-handed and a tongue roller has a father who is left-handed and cannot roll his tongue. All right, so if he can't roll his tongue, the father was, let's see what she was again. She was right-handed. He was left-handed, so that means she has to have a recessive allele because he was homozygous recessive for that because he's left-handed. That's the recessive gene, or the recessive genotype. And he cannot roll his tongue. So his genotype was big R, little t, little t. She could roll her tongue, but she had to get one of these alleles from her father. So she can roll her tongue, so we know she's big T from her mother, but she had to get one of these from her father. and they had to be small, or recessive traits or genes, because he was homozygous recessive. She marries a heterozygous right-handed man. So heterozygous, R, big R, little r, and a tongue-rolling man. Okay, so heterozygous, right-handed, and tongue-rolling. So see how I did that? If you have any questions, raise your hand or come see me and I'll explain that. So go ahead and fill this out with these. So we have big R, big T, big R, little t, little r, big T, little r, little t. And same thing, since they're the same genotypes for this side. Okay? So let's see what we get. I want you to go ahead and do this by yourself if you haven't already. There you go. I don't need to explain this any further. I've walked through this many times. If you have any question about how we got these genotypes, we just counted them from this dihybrid Punnett square, and then we turn those into phenotypes. So anything with one dominant allele is going to be right-handed and tongue roller. And only way it's going to be left-handed and non-tongue roller is if it is homozygous recessive for one of those, okay, or both. All right. So summary of Mendel's principles. Let's go ahead and write these down. The inheritance of traits is determined by individual units known as genes. These genes are passed from parent to offspring. Each gene has two or more forms called alleles. Some alleles are dominant, while others are recessive. Each parent has two alleles for a particular trait that they inherited from their parents. They pass on one allele to their offspring when the alleles are segregated. So each offspring gets one allele from each parent. And then the alleles for one trait segregate independently of the alleles for another trait. So remember why this is. We get one chromosome for each pair, 23 pairs from each parent. So we get half the genetic information. If you remember that picture that was shown in an earlier slide, we start out with a cell with 46. We double that in interphase of meiosis 1 to do 90, to have 92 chromosomes. That divides in meiosis 1 to two cells of 46 and 46, and then that divides further into two cells of 23. So we end up, that should be a 3. You get the picture. We end up with 4 cells with half the genetic material as the original cell. So each parent gives, so if we have a parent with X, the offspring will only get one of these. The other will come from another parent. So female, male, if the other parent gives a Y, and the offspring can't draw today, is, let me fix this, would be male. Okay? And so on and so forth. So I hope you understand this. Again, if you have any questions, just let me know, and I will do my best to explain further. Okay, so I kept this video a little bit shorter. Again, the next video should be even shorter. And then from there, we will go on to pedigree charts and blood typing. So if you have any questions, let me know. Once you finish this video, finish your notes, please come get your worksheet. Get that complete. It's just over dihybrid crosses. I will be posting some videos of me working those problems myself in the near future. Stay awesome. Have a good day.